as we begin to explore the actual eye itself, we have to take a look at it in terms of layers. But before we do that, let's take a look at the layers that actually are not part of the eye layer itself, and that is our conjunctiva. If you've ever heard of conjunctivitis or pink eye before, this is your bane. So it's actually part of the eyelid. If I were to take this off, and pardon our model here, that's going to kind of fall here. On the posterior side, you'll see that the eyelid behind the tarsal plates have these little pink lining with some, tarsal glands. This pink stuff is the membrane called the conjunctiva. We actually have two layers, one called the palpebrae layer of the conjunctiva, which actually is on the eyelid, and then the bulbar conjunctiva, which will actually be on the eyelid itself. What you'll find is that the eye, even though you say, oh, my whites of my eyes, are bloodshot. No. It is the palpebrae, the conjunctiva that's on the eyeball itself that has the blood vessels. Your sclera, which is our whites of the eye, do not have any blood vessels on them. So if I were to peel this away, you'd find the bulbar conjunctiva on it, and the bulbar conjunctiva will actually go right to the limbus, which is this border here. So we have already, first layer is our conjunctiva, a palpebrae portion, which is on the eyelid, and then we have the bulbar, which comes upon it. In order for me to get behind an eye, I would have to actually break it, because it kind of like folds in, and if you look at your own eye, just go to the mirror and peel your eyelid down, you'll find it goes pink all the way right to your eyelid or eyeball. After that, we have three different layers, three major layers. We actually call them tunics. The first one is what we call the fibrous layer, and the fibrous layer of the eye is the white as well as this clear portion. Mainly does this kind of structural stuff, which is the two parts, which is really nice, just the sclera, so the sclera is the whites of the eye, and then it breaks to what we call the cornea, which is a clear portion, very living. If you scratch it, it's quite painful. So if you want to torture your whatever, scratch some corneas, and that is plenty of nerves there. So we have the sclera and the cornea, and then it borders at something what we call the limbus. Inside the limbus is these tiny little holes or canals called sclerovenous sinuses, more affectionately called the canals of Schlemm, but they drain the fluid that is produced in the eye to be able to supply the cornea with nutrients, because of course if it's alive it needs some type of blood supply, but if you did blood here you would never be able to see, you just see blood, so it makes it with something what we call aqueous humor, but that's for another time. So far we have the fibrous tunic, meaning toughness for structure, sclera and the cornea with the limbus in between, and the sclerovenous sinus located in the limbus. If I were to peel this away, the next layer is going to be our vascular layer. This is one that fulfills all the kind of lots of intense blood supply, because it takes a lot of energy to be able to see. What we find is if the kind of like the sclera came around, just like this one, right? So if the sclera came around and then became the cornea, then the next one is, if I were to take the vascular layer, it actually does mainly the choroid. If you can imagine, the choroid is this purple stuff, or denoted as the purple stuff, and then it'll actually split into two layers. The best way I could do this is open it up like this and then kind of illustrate this portion here. You can see on one side of it, it gets a little thick and ridged, and on the other side it turns into the colored portion of your eye. Let's take the front portion first, and the cord goes into this area, what we call the iris, and the iris of course is your colored portion of your eye, and then there's a hole inside of it, a literal hole, and that's your pupil. So that's the black portion if you look into your own eye. On the posterior side though, we have this extra structure, and usually it's set up like this, and in a dissection you'd actually see just tiny little fibers stretching. What you'll find is you find this little thickening, and this thickening area right here, as the choroid comes out of here, you'll find this little line that demarcates the next structure. This is what we call the ora serratus. Usually it's a little more serrated. And then we get into what we call the ciliary body. The ciliary body is the place where we have a bunch of muscles in here that will help us to focus on different objects at varying lengths. So the first one is this guy here, the ciliary body. Nice big chunkaroo, has these little processes. If I were to just do this, you could hear the clicks on it because it has these little ridges on there. And so these little ridges are what we call ciliary processes. Off of the ciliary processes extend off suspensory ligaments, and these suspensory ligaments will hold this little jewel, if you would, or the lens, what is called the lens, to be able to help focus our eyes. You can imagine that I pull them at different lengths or stretch the lens, and then I can see things farther away, or I pull this or contract the muscles, and then it allows the lens to shrink up, and that lets me see things a little bit closer. So to refresh, if we did sclera and cornea, so far we got choroid on one side, to the iris and then the pupil, and on the other side it goes choroid to past the ora serratus to the ciliary body, which is the whole thickness portion that extends off ciliary processes that give off suspensory ligaments that attach to the lens. And so that is the next layer. And the last layer is actually incomplete. It doesn't actually go all the way across, and so I can identify it like on this side. You can see this little pink layer. This is all what we call the neural layer, and that is just one layer. It's called the retina, and you may hear of that. Don't look in the sun, it'll burn out your retinas, because these are where the photoreceptors are. These things are the places that will actually pick up light and change them into action potential so your brain can see the beautiful face that you have. And so here we are. There's a couple things besides just retina. There's a couple, areas of designation. You have this little white area where all the axons will actually exit, because this whole thing is made out of sensory cells, photoreceptor cells, and then they give off axons, and they all exit at this place where there is no photoreceptors. This is what we call the optic disc. Terrible, terrible misnomer, because you cannot see anything with the optic disc. Nothing. There's no photoreceptors there. So it's called your blind spot. Now don't put that on the test. Don't say blind spot. That is the optic disc. I know it's counterintuitive. You would say, oh, the optic disc sees. No. This optic disc is the blind spot of the eye because it has no photoreceptors. But there's another area right here. Hopefully you can kind of see this if I were to focus it in a little bit more. You'll find this little small area. All of a sudden, we have two different types of cells, rods and cones, and they're all distributed in the retina, but for some reason, they're not distributed evenly. Specifically, when we start getting closer and closer to this little area that is essentially directly behind the pupil, and light rays will strike it right into there, we get a lot more high concentration of cones. This area, which is like the dark circle around it, is called the macula lutea. This is a place where it's just a high concentration of cones, almost the highest concentration of cones, and then really only cones, and then deep in the center is this little divot, something we call the fovea centralis. That is the highest concentration, where the cones just pack themselves like crazy to be able to give us what we call the area of visual acuity. So you can see the sharpest in this area. When you shine light on these other areas, you'll find it get a little bit fuzzy, not because it's out of focus, but because you just don't have as many photoreceptor cells, and that's why your peripheral vision is pretty good, but it's not perfectly clear. And so let's review the last three layers here. So far we got the sclera that has the cornea, that's the fibrous layer, let's start off with here. We have the conjunctiva, which has two layers, the palpebrae portion and the bulbar one. The fibrous layer, which has the sclera and the cornea with the limbus in between, the sclera venous sinus or the canal of Schlemm found there. You have now the choroid, which is the vascular layer, which extends on the external side to be the iris and the pupil, and then on the other side you have the ora serratus as the border to the ciliary body, which gives us ciliary processes that attach to suspensory ligaments that attach to the lens, and then the retina, which then has some area designations of the optic disc, and then the macula lutea, and then the divot, which you call the fovea centralis.